Absolutely. Yeah. So if we go back to this diagram, I mean, this is precisely what's going on here. So this bacteria, again, our question of natural, of this originally, this system here we're looking at is in streptococcus pyogenes. Okay, this is one strain of bacteria, and this is just the system they use. Now, this is also in a lot of other systems. In fact, a lot of these systems are in archaea, which are this other sort of line of life that are older and whatever. And this seems to be like a form that DNA has taken for like a long frickin' time, yeah? But anyway, so streptococcus pyogenes, what the questioner just asked, that's exactly what's going on, right? This is the defense system against that sort of attack, yeah? So it's this is a virus here, right? We've got a bacteriophage, and it's going for an attack, and then this is the defense system. Once again, the other systems I talked about, restriction, modification, also, yeah? They're also just what they're saying. So yeah, that's a really good question, and that's right on the money. Do we have any other questions before that's it for me. But if you guys have other questions, or if anyone in the audience feels like asking a question, I can jump on later too. But what I will do now is I'll pass it to Bella once we get those questions, and she will tell us about her awesome work. And then we'll have a little discussion, and then we'll go get loaded. Yeah. I'm going to get loaded alone? Okay, all right, that's fine. So if it's pointing down, does that mean no one heard me for the entire time? So any questions from our audience, our viewers in Philadelphia? Yeah, there's no one from Philadelphia. Any audience questions from Milan? From our studio here in Shanghai?
I have a question.
Yeah. So feel free to use the, I don't know how.
So CRISPR, it initiates the defense system of, I'm just a little confused about the process. Can you maybe go over it once again?
Yeah.
Maybe simplified terms.
Yeah. Can you stop talking jargon crap? Like what? No. So when you say CRISPR initiates,
yeah.
So maybe you can rephrase the question because I could go through again. But what about like this? What is this here?
What is Cas9? Cas genes?
Sure. Yeah. What's Cas9?
A piece of DNA.
Right.
Yeah.
And what's CRISPR? What says CRISPR?
CRISPR is the technology that cuts out DNA.
Okay, so first CRISPR is, so CRISPR in this bacteria, the natural thing that for decades researchers are seeing and going, what's this? And let's create a stupid acronym and let's put my own, and then people keep following it. Someone asked me last time where a certain word came from, and it's always this horrible trajectory of a few egos, and then they think it's like simplifying, and it's always just nuts. This is clustered, repeating, interspersed, something, repeated, I don't know. It's a bunch of things. And all it was is they kept finding the same sequence over and over again, right? And these are so-called spacers, and this is exactly what you're looking at here, yeah? So they just found repeats, and they were like, what the heck, right? Okay, so then another paper later, and then, and then they keep doing, eventually they put together that this CRISPR system, which is now absurdly labeled, has nothing to do, there's no intuitive relationship between what it's doing and the name, right? But it's this DNA system, it's a bunch of different things the DNA makes, that we found out is a kind of genetic immune system, yeah? So we found out that it's just a way in which they can use DNA to defend itself. The bacteria can use it to defend itself. Okay. Yeah? So yeah, it's all those things, yeah? So we have, so then CRISPR, in this case, is a description of genes that do a thing in a bacteria, yeah? And more exactly, CRISPR-Cas9, Cas2, Cas5, whatever, yeah? So on the one hand, CRISPR is this description of a thing. Now, when you hear CRISPR today, you're never hearing it in that sense. You're always hearing it, as I said, in this sense, where it's CRISPR-Cas9, and it's Doudna, the important American, and Emmanuel Charpentier, the less important French researcher, who turned it into a technology. So they took one of these systems, they tweaked it a bit. You can see here, see this thing? It wasn't connected. Oh, they connected it. Okay, yeah, patent time, right? You connected a thing. No one else did that, right? So we win all the money. It's ours. And so when you hear CRISPR, you're hearing this one version that came from pyogenes and now has been changed a bit. Yeah. How do we use it? Just the way that bacteria use it. We just tweak it a bit. We use it as a way to put a gene in precisely. Why? Because this would attack. It's attacking exactly at this green, right? Not a bit outside. It doesn't cut here. It doesn't cut. It cuts exactly at the green. So that's a pretty magnificent thing. This means we can jump into your DNA, or the kid's DNA was HIV resistant genes, whatever. It's kind of stupid. But that was it can go right to that part of the genome. Now that's kind of, yeah, that's unprecedented. We didn't quite have something that was that good at finding exactly where to go. Like I said, we did have stuff that was comparable, but it just wasn't as good. Yeah. And it's an evolution. It's not this new thing that popped out of some magical head at some point. Yeah. Yeah. Yeah?
Yeah.
Yeah? Yes, and we have an audience question. Oh, okay. Yep, yep. Yes. Okay, different loci, yeah.
So my question is this CRISPR is kind of like gene aging in every cell from animal planet, or is that bacteria?
Yeah, so the first part of the question, those two things, you've seen them both, right? One is talking about what does it do in nature, what does it do before humans, we can say, or in the bacterial world, and then the other one is what we use it for, right? So they aren't editing genomes to put in what they no, they're defending themselves, yeah. I mean, they are editing genomes too, but yeah, these bacteria, right? Yeah. So, and the second question, because now we can use these and we can put them in all kinds of things, well, then now they can be in all kinds of things, right? But before, again, we came along, like I was saying, this is something in all kinds of bacteria and also in this archaea set of species, and then maybe in other things I can't remember off the top of my head, but it's certainly not something that's like common in humans, or, we have a cellular immune system, right? These bacteria only have one cell, so how do they do it? They have all these cool DNA-based immune systems, yeah. So it's more prevalent in certain... yeah. Well, and in that sense, then, it's now, it can be all over the place. So what is the key to that? What is the key to that? The key is this, okay? Because I can put a new origin of replication, I can make it work in humans, even though no human ever had this circle, because I've made it so that the human can use it. Now, I can't give you stats on the success rate of using plasmids in humans. I mean, it's nothing to write home about. But the thing is that it can work, right? And and these plasmids can work in all kinds of different organisms, right? Why? Because they get edited and tweaked and so on, right? So in that sense, then the CRISPR technique, like most biotechnologies, can be generalized to all kinds of places. We get it from one section. And we really like bacteria as like a model because they grow really fast, right? Generations really quickly. So you can do all these models really quickly, and they're small, they're easy to kind of take care of compared to other things. So if it came from there, we can put it into this, we can use bacteria as our test place, and it's very nice, yeah? But yeah. And? Another question? Okay, good. Okay, so we have to spella. Okay. So, the treat of the evening, we have Spela Petric live from her bunker, where underground she cannot be bombed. Yes? This is the last moments before the war ended, right? She was in the bunker, she was safe, Then they bomb the bunker. Yeah. Okay, so Spela Petric. Yeah, take it away. Take it away. So I mean, my quick intro is that she's just a super awesome human. She's part vegetable already. And Slovenian, based in Amsterdam now, right? Fighting the war against the West, against America from within this Amsterdam bunker. Yeah. So please, warm round of applause. Okay. Yeah, yeah, there we go. Yeah. Great. Yep, it did. Yes.
Okay, then I have one final request. I didn't realize that this is going to be streamed all over the world. I feel so dispersed.
There's nothing in Philadelphia or Milan. It's like Shanghai, and I think it's all relatives that Jeff hired. Yeah, I don't know exactly. There's a lot of people. It's Fab Lab O followers, so people who follow this Fab Lab, I guess, in Shanghai. Yeah, which may be a bit through China, but I'm skeptical. There's much else.
And I would also then maybe ask is it possible if there is a camera for the people I'm talking to?
Yes, you're pretty, Eric, but you know.
Hey. All right. Wonderful. Hello, everyone. Thanks, Eric, for this very illustrative introduction into molecular engineering. And I would like to.
So just a sound check.
Because it seems,
So we're just going to do sound here, Spella.
but it's been quite a while since I was last practicing science. And now when I'm working in the realm of art, I'm still sort of relying on the insight of science. But I'm also looking for the other meaning that emerges.
We're just doing a sound thing here, Spella. Sorry.
Okay.
So you just exit here. Okay. So people at home, you might not be able to hear this. We apologize. We're not technically equipped. So if we can get speakers in, sorry to interrupt you, Spella. Please continue. You can't hear me? Can you hear me?
Yes, okay.
A bit? Oh my God, okay. Well, I can hear you, and locally we can hear you. That's no problem, but they're just worrying about the online audience, but they have to take care of that. So yeah, just keep going. And if we have, all you guys need is a plug. Yeah, you can do this. Yeah, but you need, where's the plug? Plug. Yeah, we're in China, so. Yeah, we're here. They're looking for a chord. Yeah. Okay, so go ahead and. Okay, so you know what we can do, guys, is we can put this microphone. Yeah, we can. Okay, so now see, talk, Bella.
Yeah.
How is that? You don't know? Yeah, do this one. We can do both together. Watch this. We can do a super. No, but this doesn't. What is this for? This is RCA. Am I an alien? Okay, so talk again, Bella.
Hello. Testing. 1, 2, 3, testing.
Two. Okay. Try again, Bella.
Hello.
Okay, so we've got two microphones just working next to my speaker. This is the most ghetto budget method of amplification. Okay. Yeah? That's it? Okay. So, sorry to interrupt you. My deep apologies. Please continue. Please continue.
Okay. So I was gonna add um, add to this view of the biological, the natural and cultural, and how we appropriate phenomena in nature to use them as technology, and answer the question of human becoming plant or plant becoming human in terms of other definitions, other possible definitions that don't necessarily include the DNA, but sort of reconfigure the conception of an entity based on an entity's involvement or a relationship to the environment. So I've been working with plants a lot, primarily because I found them very strange and very different from humans. And in terms of Western philosophy, it can be said that the plant actually embodies everything that Western philosophy, Western metaphysics deems improper. So it's really about lower class of being in the Western thought, right? Because it's completely dependent on the environment. It doesn't have a centralized structure. It's very thin. There's no interiority, etc. Right? Which, of course, is not true. But this is sort of this classical value system that kind of comes from the philosophy of ancient Greece, and is still very much present. So I was thinking about different artistic experiments that I could perform to investigate this relationship between the human and the plant, the differences and the similarities. And I started off this 5-year investigation by making kind of a map that on one hand shows the differences between human and plant at various scales of organization, and you can see that it's kind of a bad puzzle, but actually the complexity gets higher from left to right. So we have the cell, the tissue, the organ, the organism, the community, and the species. And then if you kind of look at the human up till the level of the organism, we seem to be pretty much, a very tight ecology, even with the bacteria and fungi and viruses that are living within us. Then there's this level of the surface, and we start thinking then about the community and the species. Whereas, hello? Yeah?
Go ahead. Yeah, great.
Can I continue?
Yeah, please continue. It's awesome. Yeah. You've been a rack on.
Okay. So whereas in the plant, there's a lot more plasticity, so you can cut off a leaf and stick it into water and it will grow roots, and you will have another thing that might be considered an individual. But in fact, plants tend to grow like that with a common root system, and then at one point maybe get separated, and then you have two individuals. So there's this conception of identity is formulated a bit differently, right? So I figured that actually a lot of this tension, right, and this differentiation between the human and the plant arises at this level of the individual. On the other hand, another thing that kind of frightens me still is the role of people in this network and the wider social organization that is so permeated by algorithms at this moment. And I think we are very much not only taking on properties of the plant, but it's kind of, we are put in a position, a similar position that the plant has been up till now. So it has nothing to do with the actual bodies. It has to do with the social relationship. So, but we can talk about this at the very end. I would like to start by just briefly mentioning this Project Strange Encounters. When Eric was talking about what it would take for a human to become a plant or the plant to become human, on the level of cells, I really wanted to see what happens when you kind of put human cells and algae cells in a petri dish, make the environment really nice for them both, and see just what happens. And surprisingly, or not surprisingly at all, this is sort of a micro-performance, what happened was that we got a formation of a new hybrid cell. So this is, you can see here the cancer cells that are attached to the bottom. And here in these brown cells with the green in the center are the cancer cells that have ingested the algae. Now, I wasn't able to follow this through what exactly happens to this algae in the cancer cells. But, and initially I sort of dismissed it as, well, the cancer cells are anyway hungry, and they would ingest everything that could possibly be nutritious. That's what cancer cells do. However, I much later read an article that in biogenesis, which is basically the way that eukaryotes were formed by certain cells ingesting other bacterial cells, but not digesting them. Instead, they became internal partnerships and this form that is complex and that benefits both partners that at one point become inseparable. Well, that this occurrence is commonly observed as long as you have different species in the right conditions growing together in the laboratory for a long enough time. So I like to think that perhaps if I had followed this through, that we would maybe have, an occurrence of a human plant and this strange hybridity of ingestion, of making another type of being with plants. That is not about understanding genetics as a toolbox to play with, because anyway, it happens in nature and there's many different ways of approaching it. The other thing that we also would maybe or should consider is the fact that genetics, and we like to use this similar a lot, is like information, and the DNA is code and it's sort of at the heart of every organism. So it, culturally takes on this icon of the soul, but not in a material sense, but in the informatic sense. So while we are so obsessed with DNA, we are kind of ignoring all sorts of other processes that make meaning, right? So like DNA is a code, it connects basically one type of molecule, and DNA, RNA, with a protein, and the protein then takes part of a metabolism. And so all this seems to work, whether you look at it as a cybernetic system, as an autopoietic system, or, as an ecological system, because the material the material has meaning to different interpreters, right? So even though we don't understand all living processes as a kind of language, there is evidence to believe that it actually is. So it's not, there's sort of a duality to all the constituents of living organisms, which is they perform like a material function, right? They're made out of material, but they always carry also meaning in the context, right? So what is being more and more discovered is that things that were in the cell and appear in cells and were thought to have structural functions are actually codes, right? They're all codes. Like microtubules in neuron cells are supposed to be significantly affecting the pathways of neuronal transmission, which might be one of the reasons why memory can be stored quite easily, even though there's a physical modification involved. So this is a whole area of, I would say, fringe science that deals with it. It's called biosemiotics. And it really extends this notion of science to biological processes. So this is also something that I wanted to explore, this kind of connection between human and plants in terms of biosemiotics, which would then provide some sort of living, some sort of real material connection, a language, if you like. And I don't know if you know this, but clover, which is, like the four-leaf clover, the popular food of rabbits. But it's also, it has a way of maintaining a certain population of rabbits because, rabbits like to have sex. So they multiply like rabbits given the opportunity. But the clover has mechanisms that when it's raised, like when it's eaten and the rabbits are peeing on it, it recognizes, oh shit, it's the rabbits again. And then it starts synthesizing a hormone that acts like a contraceptive pill for the rabbits. Therefore, it controls the population of the rabbits. And